All right, let's jump right into this explainer. We're going to treat the history of life on Earth like an ancient detective's notebook. Now, since you're already familiar with the broad geological timelines, we're skipping the surface level stuff today. Instead, we're zooming straight into the hard, specific molecular and genetic data. We want to see exactly how macroevolution actually happens, looking at the raw chemical evidence behind life's biggest transitions.
So, here's our roadmap for today. We'll hit the molecular origins of life, dating the fossil record, endosymbiosis and cellular complexity, evo-devo and macroevolution, and finally, evolution as molecular tinkering. Let's get to it.
Section 1, Molecular Origins of Life, or How Chemistry Became Biology. We're starting way back in the Hadean Eon. How do we go from absolutely nothing living, just raw elements, to a fully metabolizing cell? Well, scientists break this massive transition into 4 distinct, testable stages. First up, the abiotic synthesis of small organic molecules. Think amino acids and nitrogenous bases. Next, those tiny molecules join up to form larger macromolecules, like proteins and nucleic acids. Third, these macromolecules get packaged into protocells. These are basically little membrane-enclosed droplets that maintained an internal chemistry totally different from the chaos outside. And the fourth stage? The origin of self-replicating molecules, which literally made biological inheritance possible.
Now, to test that first step, you've probably heard of the classic 1953 Miller-Urey experiment, right? The one simulating the early atmosphere. Well, it actually yielded a pretty low variety and mass of amino acids, but the real game changer came much later in a 2008 reanalysis. Researchers took modern, highly sensitive mass spectrometry gear and analyzed some old sealed vials from a different Miller experiment, one where he specifically simulated a volcanic eruption. And wow, the results were staggering. They found an immense variety, over 20 different amino acids, and a mass of over 200 milligrams that formed entirely abiotically. This is absolutely crucial because it proves that localized reducing conditions, like those dense, chemically heavy clouds spewing from early volcanoes, were basically giant molecular factories perfectly primed to churn out the building blocks of life.
So, jumping to that fourth step, inheritance, we have to wrap our heads around something pretty profound. Before DNA ever existed, life was most likely an RNA world. The absolute first genetic material was probably single-stranded RNA. These early RNA molecules could fold into a variety of highly specific 3D shapes based entirely on their nucleotide sequences. And some of these, called ribozymes, actually acted like enzymes and could make complementary copies of themselves. Now picture this: abiotically produced vesicles packed with montmorillonite clay. That's a soft volcanic mineral that actually chemically accelerates the self-assembly of these vesicles. When these little droplets naturally absorbed the self-replicating ribozymes, boom, they crossed a major threshold. They became the very first metabolizing, self-replicating protocells, literally chemistry becoming biology.
All right, Section 2, dating the fossil record, the radiometric clock.
Okay, so we have these early cells, but how on earth do we accurately date their fossils across billions of years? The magic number here is 5,730. That is the exact half-life in years of carbon-14. See, while an organism is alive, it's constantly absorbing carbon, including the radioactive isotope carbon-14. But the second it dies, that intake stops, and that carbon-14 begins to slowly, predictably decay into nitrogen-14. By measuring the ratio of carbon-14 to stable carbon-12 in a fossil, we can precisely date specimens back to about 75,000 years. But wait, for macroevolution, we're talking billions of years, right? For the really old stuff, we have to use indirect methods. We date the surrounding volcanic rock layers using radioisotopes with massive half-lives, like uranium-238, which takes about 4.5 billion years for half of it to decay. That effectively traps the fossil in a precise, undeniable geological timestamp.
Moving on to Section 3, endosymbiosis and cellular complexity, or the Great Merger. So how did life level up from simple prokaryotes to the incredibly complex eukaryotic cells that literally make up your body today? If we map the molecular milestones, there's a dramatic shift driven by sheer chemistry. For a long time, prokaryotes ruled the Earth, starting around 3.5 billion years ago.
But then, roughly 2.7 billion years ago, the oxygen revolution hit. Cyanobacteria figured out how to split water molecules during photosynthesis, and free oxygen just flooded the atmosphere. Now, since oxygen aggressively attacks chemical bonds, this actually doomed countless groups of anaerobic prokaryotes. It was highly toxic to them. But it cleared the stage for a new breed of oxygen-using eukaryotes, which violently burst onto the fossil record around 1.8 billion years ago. And we know exactly how those eukaryotes formed, through serial endosymbiosis. The molecular data here is just undeniable. The idea is that a large host prokaryote engulfed a smaller aerobic bacterium, which eventually turned into the mitochondrion. And the proof? It's staggering. Both mitochondria and plastids have inner membranes that are completely homologous to living bacteria. On top of that, they replicate by an independent splitting process that looks remarkably like bacterial binary fission. They even have their own circular DNA with no histones. And they contain bacterial-like ribosomes. The undeniable molecular signature of ancient bacterial ancestors is quite literally hiding inside your own cells right now. How wild is that?
That brings us to Section 4, Evo-Devo and Macroevolution, Genetics of Body Plans. So once we hit the Cambrian explosion and life starts colonizing land, how did animal body plans diversify so insanely fast? The answer is in evolutionary developmental biology, or Evo-Devo, where tiny genetic tweaks trigger massive morphological shifts. A perfect, all-inspiring example of this is altering developmental timing, specifically something called paedomorphosis. It's caused by a broader process called heterochrony, which is basically an evolutionary change in the rate or timing of developmental events. In paedomorphosis, an organism's reproductive development massively accelerates compared to the rest of its somatic organs.
I want you to put your evolutionary detective hat on for a second. How could a whole body morphological shift happen with just a mutation at a single genetic locus? Just look at the axolotl. It's an aquatic salamander that grows to full adult size and reaches sexual maturity, but it keeps its juvenile, larval gills its entire life. Recent molecular evidence actually points out that a change at literally one single genetic locus was likely enough to completely alter its developmental timeline. One tweak, huge result.
Let's look at another striking piece of data, this time on spatial patterns. The real secret to these massive body plan changes lies in master regulatory Hox genes. Scientists wanted to figure out how six-legged insects evolved from crustacean ancestors that had tons of legs. So they cloned the Hox gene called UBX from a Drosophila fruit fly and an Artemia brine shrimp. The insect UBX suppressed 100% of limb formation in the fly embryos, but the crustacean version, it suppressed only 15%. By creating hybrid genes, researchers nailed down that swapping just a handful of exact amino acids in the nucleotide sequence was enough to completely shift the insect gene into overdrive, actively and aggressively suppressing all leg formation in the abdomen. That is molecular power. But here's a really fun scientific mystery for you. What if the DNA coding sequence itself doesn't change at all? Let's test this out on the three-spine stickleback fish. Now, marine sticklebacks have these large ventral spines to fend off ocean predators. But populations that got trapped in predator-free lakes, they completely lost those spines. So researchers asked a fundamental question. Did the actual genetic coding sequence of the PITS1 developmental gene mutate and break, which we'll call hypothesis A? Or was it just a change in the regulation of that exact same gene, hypothesis B?
Well, let's look at the molecular proof from in situ hybridization. They used PIX1 DNA as a fluorescent probe to track mRNA in the fish embryos. And guess what? They decisively proved hypothesis B. The 283 amino acids of the PIX1 protein are 100% identical between the heavily armored ocean fish and the spineless lake fish. The coding sequence is perfectly intact. However, a highly localized regulatory mutation in a non-coding DNA region, called the PEL enhancer, simply flipped the gene off exclusively in the pelvic region. So a massive, life-saving anatomical change happened not because a gene broke, but simply because a localized genetic switch was flipped.
And finally, Section 5, Evolution as Molecular Tinkering, or Repurposing the Past.
This brings us to our final core concept. The biologist Francois Jacob famously said that evolution is like tinkering. It's definitely not goal-oriented engineering where you start from scratch with a pristine blueprint. Nope, it's blind molecular tinkering with whatever parts happen to be lying around. We see this all the time through a process called exaptation, where existing structures are just repurposed for entirely new functions over millions of years. Take the squid eye. It's incredibly complex, practically camera-like, but it evolved incrementally from a super simple patch of light-sensitive cells found in a common ancestor.
Or consider your own hearing, which is honestly an even wilder data point. The delicate hammer and anvil bones in your middle ear didn't just pop out of nowhere. They were co-opted. They actually evolved from the heavy articular and quadrate bones that made up the primary jaw hinge in our ancient reptilian cynodon ancestors. As those mammals evolved a totally new jaw hinge, those massive original bones gradually detached, shrank down, and migrated inward for the sole purpose of transmitting sound waves.
All right, I'll leave you with this provocative thought to analyze, and I want you to really let the weight of this sink in based on today's explainer. Imagine the myxoma virus suddenly sweeps through and kills 99.8% of a dense rabbit population. It's basically a near total extinction event for them. Think about all the microevolutionary mechanisms we just systematically broke down. What exactly is happening at the bare molecular genetic level to that tiny 0.2% that somehow managed to survive? And on the flip side, what evolutionary trend is going to aggressively emerge in the millions of viruses that absolutely depend on living hosts to survive? The entire history of life on Earth is written in these exact survival mechanisms, heavily documented in the mutated genes left behind. Keep tinkering with these concepts, and I'll catch you next time.